Everybody, welcome to the Fate and Exposure Assessment of Nanomaterials session. And this session will be contributed to by 4 people: myself, Teresa, working at Heriot-Watt University in UK, Socorro Vazquez, working at LEITAT in Spain, Joris Quik, working at RIVM in the Netherlands, and Sam Harris, also working in UK at the Centre for Ecology and Hydrology. So this is the way we're going to work this session. I'll start a quick introduction to the topic, and this will be followed by Sokus talking about likelihood as well as routes of exposure to nanomaterials. Joris and Sam will focus on modeling. Joris start introducing the topic, and Sam providing a little bit more depth to the complexity of this field. You will be able to practice for yourselves the concepts that we'll be describing and discussing, and then we'll be discussing the results of your exercise and wrapping up with the lessons learned. So you've read the exercise. As I said, will allow you to test some of the concepts we're going to introduce here. So hopefully you're able to follow instruction and download the model without any problems. So why nanomaterial first and exposure assessment? Why should we be concerned with that? Obviously, nanomaterials are going to be quite different from conventional materials. So they are, as we'll say, they are particulate material, and they are small materials as a starter. So they're not going to disperse as well as conventional chemicals, and because they're so small, they obviously will have a different fate when compared, and potentially impact it when compared with larger materials. So here you can see there's such a diversity in terms of size, for example, in terms of shape, in terms of actual composition, also surface covering. Different surface coatings, for example, are very much in practice in the available nanomaterials. So very large diversity, and so it is important to understand what happens to them in the environment, and also how they might affect. So you can see here they are incorporated in a variety of products. In order to do so, you will need to produce them, you need to incorporate them in these materials, and then during their use or after their use, they might end up in the environment directly through fragmentation, for example, or if they are part of sunscreens, they might end up in the water, etc. But often they are products that end up disposed in some way into the environment, and then they might leach again. So it is important for us to understand the different steps and how the diversity of nanomaterials may actually lead to different types of fates and exposure. Of course, the manufactured nanomaterials are just a part of nanomaterials might exist in the environment. Some of them, as I said, will end up in the environment at the end of life or during use, but you might also find different types of accidental spills leading to exposures, or those products ended up in the environment and leading to different types of the exposures. On the environment, so I've already talked about the diversity. On the environment, they might suffer or go through a variety of transformations. This might depend on the receiving environment. If it's salt water, is for lower pH or higher pH. If it's a higher abundance of organic matter, for example, in soils or waters, all of that will affect the transformation these nanomaterials might go through and where they might end up. Some of these transformations might actually be mediated by organisms, such as microorganisms, for example. So understanding these and some of the modeling that we will cover in the next presentations will help us to elucidate some of those steps. And of course, very importantly, is to understand what will happen once you actually have all these transformations. What will happen when organisms get into contact with these nanomaterials? This might be in soil systems, for example, where you have plants, and this might have links with agriculture and maybe inputs into the human food chain. You'll have other types of organisms living in these systems, such as worms, which provide a very important nutrient cycle, for example, in soil systems. Will they be affected? In the aquatic systems, you might have impacts or at least encountering primary producers, secondary producers, and you might have the whole food chain becoming in contact with these available nanomaterials, which would have been transformed somehow. This might be impacting over different generations, as you can see here. There's a possibility of that. And of course, when nanomaterials get into the organisms, they might lead to a variety of effects, which might be impacting respiration, for example, might impact detoxification, might impact might lead to genetic toxicity. So a variety of effects might take place within cells, within tissues, within organisms. So understanding fate and effects of fate and exposure to nanomaterials is instrumental to allow us to assess what hazard might take place when organisms are put in contact with nanomaterials in the environment. So this concludes my presentation, and I'll pass on to Soko for the next step in this topic.
Hello, my name is Socorro Vazquez Campos. I'm from LEITAT Technological Center, leading the group of human and environmental health and safety in this research center. And today I will talk about release and exposure along the life cycle of nanomaterials and nano-enabled products. I would like to start with an introduction of the terminology. So the term release refers to the liberation of a nanomaterial during a natural or technical process along the life cycle that takes place along the life cycle at any stage of the life cycle of the nano-enabled product or nanomaterial. Then the emission is the transfer of this liberated nanomaterial or substance to a compartment where it can reach a receptor. And this emitted or liberated nanomaterial then suffers the transmission towards the receptor, and then the exposure is when the nanomaterial come into contact with the receptor, which in the receptor can be the human or the environmental species, and possibly through any route of exposure. During this transmission of the liberated nanomaterials, they can happen a lot of transformation and the transport of these nanomaterials in the compartment or in the environment, and this is known as the fate processes that take place during this transmission step. All these events are linked to each other, and any of these events cannot take place if the previous one didn't happen before. So they are subsequent to each other. So then there is a release of nanomaterial, then an emission, and these emitted particles end up to be the exposure particles that go into the target receptor, human or environmental species. So continuing with the introduction, I would like to explain on the life cycle perspective that nanomaterials are generated first during a process that takes place to synthesize these materials. Then they can be manufactured into to be ending up being an article or a product, and then can be used during the by the consumers, and this is during their service life is this use phase stage of the life cycle. And then when they are already used and their service life is finished, then it comes to the end of life processes, which means either waste the product or the article or recycle this article into something else. And all these life cycle stages, during those stages, many processes can take place that can modify and can influence the releases of that of these nanomaterials in each of the stages. Here are some examples of these processes that we also call it activities that they take place during the synthesis, manufacturing, use phase, or end of life. And during this, we can monitor the nanomaterials releases, but of course, there is some of the properties of these processes will influence this release during the life cycle stages. So then, of course, during these life cycle stages, we also have different receptors that can be exposed to these release materials, and these are human and environmental species that that are in contact with this during the different stages. So during the synthesis, manufacturing, use, and end of life, you can have some workers, consumers, and that can be exposed to release materials during these processes. And then we also these emitted particles can be liberated in any of the environmental compartments if the processes they take place outside or in contact with any of the environmental compartments, which can be via the air, water, soil, roots, and ending up in the different exposure of the different species. Then how can we determine these releases along the life cycle? So we have to come up with the release rate, and we have to determine also the form of release. So if it's a nanomaterial, is an ionic form of the nanomaterial, is in the form of composite material, and then also to determine who is going to be exposed, so who will be the receptor, following which exposure route these release materials will end up to be in contact with these receptors. And for that, we have to do different monitoring of these releases for the purposes of quantifying, but also for the purposes of determining the properties of these release materials, to be able to come up with all this information that we will need to finally determine the exposure. And for that, we have some possibilities of direct monitoring of these release materials during the activities or the processes that take place during the life cycle. But we also have to use some simulation methodologies that we can use as accelerated processes to simulate the real scenarios where these processes take place and simulating their characteristics to be able to control this monitoring of the release materials. For that, I showed you here some of the examples of processes that are simulated in the lab, some of the instruments that we are using, instruments in which we follow standard procedures to determine what is the release form that gets it's emitted during the processes that take place during weathering, for example, during washing, during the use phase, these processes, abrasion processes, leaching and incineration that will simulate the end-of-life processes. So we have several processes that they can be lab simulated under accelerated conditions and following standardized methodologies. And of course, together with this and really linked, we do have to have the analytical methods to monitor the properties of the release materials, because the complexity of the release materials can be really high, because these nanomaterials can be embedded in different articles or products depending on what are their applications. So we need a combination of techniques, and here are some of the techniques that we are using in combination to be able to determine the properties. And here are some examples of quantifications and identification of these engineering nanomaterials emissions during the different life cycle stages. And these are examples of emissions that could end up in the environment, so at the different life cycle stages. And these examples have been run during different projects, Calibrate and NanoFASE, and the release rates have been determined. So the data that we could get out from these experiments is showing here how we can get the release rate, release form, and the compartment where it will end up. And this information is all requested by the model, so we have it produced. And what we can learn also from all these studies is that there is a lot of determinants that are, we have to consider in terms of influencing the release and exposure. And those determinants are mainly related to nanomaterial characteristics, the process characteristics, and also the if there is any system dependent variables, which means if we have risk management measures in place, the emissions will be reduced, and then there will be some calculations due to this needed to come up with the real exposure factors that we have to consider for the concentrations and identification of the release materials. Then this information, of course, is useful for read across and grouping, and all this data that is generated is, well, of course, compiled in release and exposure libraries. We generate templates for the whole community, so these templates are available. they've been generated in gracious projects, and now they are openly available for used in by the whole community in other projects. So we have these exposure and release templates with data in to be used for the models. So the key messages or the highlights highlighting the key steps for assessing release and exposure of nanomaterials and enabled products are the identification of the life cycle stage, so the processes and activities that take place in this in each of the life cycle stages and what are relevant for the release and exposure, identification of parameters driving the release, and that means the properties and the characteristics of the nanomaterial, the characteristics and properties of the product matrix into which is the nanomaterial incorporated, and then also the type of activity and process that this nanomaterial will be going through during the stage of the life cycle. And then from all these, you generate a lot of data that is should be generated with standardized procedures and methods, and then collect this data in a harmonized way using these templates of release and exposure. And then the data will be used for modeling purposes, for validation, refinement of the existing models, and also for generating new predictive models for exposure assessment. So these are the highlights, and I thank you all for your attention and open for questions.
Hi, my name is Joris Quik, and I'm here to present a presentation on multimedia fate modeling, specifically on the model SimpleBox for Nano. Now, who am I? My name is Joris Quik. I did my bachelor and master's at the Wageningen University, and my PhD on aquatic fate of nanomaterials at the Radboud University and lab work at RIVM. I did also a postdoc on aquatic fate modeling of nanomaterials at Wageningen University. Currently, I'm employed at the RIVM, which is the Public Institute for Health and Environment, currently known for all the corona advice they give and so on, and research on that topic. But I work on environmental exposure assessment, mostly nanomaterials, but also other substances, microplastics, and so on. Also, I work on projects related to circular economy, sustainability, how to assess or those types of topics, specifically in the overlap with safety and risks of chemicals and compounds. Also, these are my views, not particularly those of the RIVM. So I'll be talking about a little bit introduction about exposure modeling, and then going into the details of SimpleBox for Nano, how it works, what are the input parameters, the output parameters, and some examples of the model applications, some studies using the model, just briefly. And then in the end, there will be some slides and links to go to some further information if you really want to use the model or know more about it. So before we get into the modeling, when you want to know the exposure to a certain substance, it starts with an emission somewhere. You make a model, you use a product, these type of things. Then a substance can get emitted, or in this case, nanomaterials, to the environment. There they undergo different fate processes. They end up somewhere where there are organisms which are exposed. And if this exposure is too high, meaning you compare it to an effect threshold, to the safe level, then there is a risk, of course. Which is not always the case, but it's important to know the exposure to assess the risk. Now, Actually, you can measure, of course, your concentration, which is, of course, done very often, monitoring different substances, monitoring their spread, and but that's, of course, after it's already been emitted. That's when you can. But sometimes it's very handy to know it beforehand. For example, if you want to allow a certain product on the market, or you think you can use a new type of material, it's very convenient to know beforehand what your exposure might be when you use it to a certain extent. So this is why modeling becomes quite interesting to apply this in prognostic risk assessment. For example, here we have a graph of SimpleBox for Nano outputs on the left, which are calculations, predictions. This type of graph you get when you apply the model probabilistically. And of course, this is for microplastics, but of course you can also measure these concentrations of microplastics already because they're in the environment. And then you can compare them to the modeled ones and, let's say, start validating the model and so on. But this is not always as easy, for example, for some that are, of course, not out there yet. Now, what is SimpleBox for Nano? So it's actually a screening level model. It actually calculates more background concentrations in large compartments, well-mixed compartments. In this case, focused on the environmental fate of nanomaterials, describing the process affecting fate. And so it predicts exposure, the concentrations in air, water, soil, and sediment. And it is built in a way that it has nested scales. So you have a global scale within which there's the continental scale and the regional scale. And in theory, you could add a local scale, but this is not part of the tool as it is right now. So SimpleBox actually was an existing model. It's actually applied within regulatory frameworks such as REACH, and SimpleBox was the basis for the EU's, the European Union System for Evaluation of Substances module on environmental fate. And so this makes it quite interesting to also make this fit for use with nanomaterials. Now, why would we need to make this model fit for nanomaterials? Well, the conventional SimpleBox, or SimpleBox 4, is of course fit for use with chemicals, but these are dissolved, for example, organic compounds, and they distribute themselves between the gas, liquid, and solid media. And this is described using thermodynamic equilibria with partitioning coefficients and so on. So for nanomaterials, it was found that actually because they are a particle, they're solid, they are thermodynamically unstable, so they will not follow partitioning coefficients known for, let's say, other compounds. Also, dissolution plays a role because they dissolve. For example, a nanomaterial is transformed to another form. Let's say you would have a dissolution of a silver nanomaterial, and you would get dissolved silver through dissolution, which is then the conventional form of the chemical. And there are other transformation products which are important, such as heat agglomerates, because they tend to aggregate these nanomaterials and stick together. So this adaptation resulted in different algorithms required, and one of the transport processes that needed to be changed was deposition, because particles have a mass, they actually can settle out of, for example, the water phase, and the deposition from air to soil and water is also different. So also the solution, as I said, plays a role, and also the attachment to other natural particles. These were the main fate processes that were changed or added to the model specifically for use with nanomaterials. The other way was we could not use rate distribution coefficients, so we had to use rate constants, which describes the flow of the material from one form to the other, the rate. So in the end, when we look at transport, deposition was the main process that we changed, but advection, movement with a body of air to the next, through wind or flow of water downstream, this is still the same. Also related to transport between different compartments, there's no evaporation from, let's say, water or soil to air. There can be some resuspension, but this was found negligible and not included in the model. So these transformation processes are a bit more interesting. We included two types of aggregates. So basically, we call that heteroagglomeration when a nanomaterial sticks to another particle, a natural occurring particle, because this is ubiquitous, right? We have aerosols in air, we have sand and particles in soil, and we have other suspended colloids and so on in water. So they attach to those, and we divided them in coarse heteroagglomerates and smaller colloidal heteroagglomerates below 450 nanometers. Then, of course, you have dissolution, where the specifically metals would dissolve. Of course, degradation can also be a process, particularly if you look into organic type of nanoparticles. Now, just to show a little bit of what is behind the model, I just show you how. So the model is basically built up of these rate constants, and dissolution. For dissolution, this is how the function would look, where you have change in concentration over time dependent on the dissolution rate constant and the concentration, right? For heteroagglomeration, it becomes a bit more complicated because there's not a heteroagglomeration rate constant per se. This is dependent on this big K, which is the collision frequency, the attachment efficiency, which is the alpha, and this is the efficiency and the frequency of collision between the nanoparticle and the natural colloid, right? They would meet, and this is the frequency that they would meet, and then the attachment efficiency is the efficiency at which they would stick together for the rest of their life. No, so these are two equations, one describing the concentration, change in concentration in time of the nanomaterial, or the change in concentration in time of the heteroagglomerate. Now, basically what then the model describes are three different particulate concentrations, those of the nanomaterial, those of the natural heteroagglomerate with the nanomaterial attached smaller than 450 nanometers, which when you look at the bioavailable concentration as a cutoff of 450 nanometers would be the combination of the two. And you have the total particulate concentration, which is also including the coarse fraction of heteroagglomerates. And then, of course, we also have the concentration of dissolved compounds. Now, the inputs required to run the model, they require an emission rate, so to the different environmental compartments, such as air, water, and soil, for a nanomaterial. We also require physical chemical properties of the nanomaterial. These are the size or the diameter or radius of the particle, the density, the attachment efficiency, the dissolution rate, and the Hamaker constant. The Hamaker constant is related to the interaction between a particle and another particle in water. So, of course, this is done for a certain landscape scenario, right? And this is, for example, the EU with a certain catchment, the Rhine catchment, which is, let's say, the default scenario in SimpleBox. So you don't really need to input a lot of these numbers. There are already defaults given for you. But these characteristics that were changed for the nanomaterial model, SimpleBox for nano, were that we needed some properties for the natural particles, such as size and density. Furthermore, these are all properties of the three different scales. We have properties of the lake, river, and seawater, three types of water, and agricultural, natural, and other soil. So another characteristic of SimpleBox is that it calculates the concentrations in steady state. This means that the mass flows in between all the compartments are in balance, right? They're the same. There's just as much going into the compartment as going out. And this is done by a simple calculation. Actually, you take the inverse of the matrix of all the rate constants and degradation rates of the compound, take the inverse and multiply that by the emission, and then you have the mass in each of those compartments of that substance or nanomaterial. This formula, you can see that actually the mass and in the end the concentration, which is the mass divided by the volume of the compartment, is linearly related to the emission rate. Now, what is happening in steady state, it is not immediate, right? It can take time to reach this steady state. And for that reason, there's also a dynamic module, which you need to do currently. The easiest way to use the module is to do it in R with some R scripting. But what you get then is, for example, a calculation for each time point, and you can see what the concentration buildup in time is in each of the compartments. And on the right-hand side, I just want to briefly show this example, and this is again for microplastics actually, so not nanomaterials, excuse me for that, but they're particles nonetheless going with the same processes as nanomaterials. So what you see in the top here is that for example, the free microplastics reach a concentration, the steady state concentration within days even. But then the agglomerates reach a bit later. And what you see here actually is that the marine run here does not reach steady state. Now, what I didn't tell you is the difference between the top and the bottom, which is the top ones are for microplastics that actually float and without any degradation processes and so on, or very high degradation processes in seawater. You can see that it takes more than 1 million years actually to reach this steady state, which is quite weird. Where if you have a fragmentation process, also these very light buoyant microplastics actually degrade, and you can see that within 100 years, almost 100% of the steady state is reached. So this is why it can be important to look at the dynamics of the system, and this is provided also by the model, although it's currently not. There are some software issues where we used to have a macro that actually did this, but right now it needs some manual tweaking in R to use. But the steady state concentrations are as simple as using an Excel spreadsheet to calculate. So this is another example of use of the model, or studying what the model actually means in terms of sensitivity to certain key parameters that describe fate of nanomaterials. So in this case, related to the dissolution of any type of nanomaterial actually. So what Joris Meesters did, he just varied these parameters and looked at the change in concentration that gave. And he saw that for dissolution rates above 10 to the power minus 12, the PECs were sensitive for the total range of particulates, so bigger and smaller ones. But he did the same for the attachment efficiency, which I briefly mentioned before, which is related to this heteroagglomeration with the particles, between different particles. So the nanomaterial versus the colloid or coarse. And you can see that it's sensitive to the whole range of tested attachment efficiencies, although the effect is different. In some compartments or some concentrations, the concentrations are increased when you lower the attachment efficiency. In others, it is the opposite. And these critical ranges are actually important to be measured, right? This is what you need to know. And this is also related to the test on how sensitive your measurement methods should be. And this is helpful for regulating nanomaterials and their. In the end, SimpleBox for Nano is aimed to be a regulatory relevant screening level multimedia fake model for nanoparticles. We have learned a lot from different projects in which we worked on the model. We have some more work, which I did not all present here. We continue to work on it mostly now in relation to microplastics. Of course, you should be aware that for predicting local concentrations, there are some adaptations required and some additional estimates of a local predicted incremental concentration, which the current SimpleBox for Nano does not provide. So I thank you for your attention. Any questions or inquiries, please, my email address is given here. I cannot guarantee immediate response, but please feel free to contact me if you need any further help. So There is already an existing webinar actually from a few years ago, which might be quite helpful. They also provide some a bit longer talk on how to use the model, and also some case studies, which are also available in the model itself. So it's quite handy to actually have the model, click through it and look at that webinar, I think, if you really want to use it and still feel you need some help. The model itself is available for download on the RIVM website, so the nano version and the conventional version. GitHub, there's the link. You can actually find some of the scripts that are relevant for using the dynamic module, and there are several papers on the use of the model, the report. So please have a look at these. Thank you again for your.
Hi folks, my name's Sam Harrison and I work at the UK Centre for Ecology and Hydrology. So I'm an environmental contaminants modeller, so I spend a lot of my time writing spatial and temporal models of how potential contaminants such as nanomaterials move around the environment. And today I'm going to talk to you a bit about a model that I was the lead developer on, and that's the NanoFASE model, nanomaterial fate and speciation in the environment. So the NanoFASE model is a spatiotemporal multimedia model. And what that means, so spatiotemporal, that means that it predicts things with spatial distribution by splitting up a geographical region into a number of grid cells. And it predicts things with temporal resolution by splitting a time period into a number of time steps. And it's multimedia, so what we mean there is that it covers different environmental compartments. So it covers soils, water bodies such as rivers and lakes, and bed sediments. We've been talking a lot about exposure assessment today, and so before we dive into the model itself and have a look at the model and its results, I thought I'd reflect on this question, which is basically why do we need a spatiotemporal model as part of an exposure assessment? So we've already seen from Joris SimpleBox for nano model. So the question is, why do we need something like the NanoFASE model over and above SimpleBox for nano? And this idea of a tiered exposure assessment is one way in which we might need a spatiotemporal model. So if you have a look at this flowchart on the right hand side of the slide, this is a simplification of how a tiered exposure assessment could work. And the idea here is that you start off with the simplest but most conservative calculation of exposure and thereby risk that you can make, and that could be a simple back of the envelope type calculation where you make some really conservative assumptions like all of the nanomaterial released goes to one compartment and none of it transforms into other chemical forms, and you calculate a concentration based on that. And if you have some idea of hazard, you can then calculate risk from that. And because this is really conservative, if you calculate risk and it says no, it's not risky, then you know that you've kind of covered all bases and you don't need to do anything more. But if that simple calculation comes back and says, well, actually there might be a risk, then you can go on to a more realistic model. And one example of that, as we've just seen, is SimpleBox for nano, and so on and so forth. And the idea here is that basically as you move through these tiers, you're decreasing the conservatism, but you're increasing the realism of the model. And so as you get towards the higher tiers, like something like the NanoFASE model, you have a more realistic picture of what's going on in the environment and what the real environmental risk from your nanomaterial is. But you don't want to start these really high tier models because they're complex and they're difficult to run and they require a lot of data. So I'm just going to quickly show you a couple of results that show you the types of things that you can get out of one of the higher tier model, something like the NanoFASE model, over and above screening level or simpler models. And I'll do so using an application of the NanoFASE model to the Thames catchment. And here we're splitting the Thames catchment up into 5x5K grid cells, looking at years from 2000 to 2020. And this is for nanotitanium dioxide. Because it's a spatial model, we can answer questions such as where are the hotspots? So we can split up a geographical region into spatial grid cells like we have here. And what this map here is showing is the Thames catchment. And the more yellow that a grid cell is, the higher the PEC, the predicted environmental concentration, is in the soils in that grid cell. So we're seeing the soil nanomaterial concentrations. And this is at the end of 2020, so after our 20-year run. And the conclusion we can draw from this actually is that the main determinant of the spatial distribution of nanomaterials is land use. So a key way that nanotitanium dioxide gets onto soils is through the application of sewage sludge, and that's higher in agricultural areas outside of our urban areas, which is why you see low concentrations in the urban areas like London and higher concentrations in the rural agricultural areas. And because it's a temporal model, we can look at the dynamics, and particularly we can look at the dynamics of more dynamically interesting compartments such as rivers and estuaries. So here, this graph on the right, I'm showing you an illustrative river reach and an illustrative estuary reach. And basically the main conclusion to draw from this is that the riverine nanotitanium dioxide concentrations are strongly influenced by the hydrology of the system. So you can see in the river all of these peaks, and those peaks occur during the drier months of the year. So drier months means that rivers are flowing more slowly and they're generally smaller, and because they're smaller they have less volume, which means that nanomaterials are more concentrated. Estuary, on the other hand, doesn't show as much a dynamic trend, and it actually shows a bit more of a trend that follows the emissions trend, which is shown at the top there. And the reason for this is basically because water can come in and out, up and downstream in an estuary reach, and this upstream motion has the effect of almost trapping nanomaterials in place, so you're not getting as much of a flushing through of nanomaterials that you do in river systems. So spatiotemporal models like the NanoFASE model they give us lots of information, but this is at the expense of being fairly difficult to use. So because they're spatial, you need spatial data, you need temporal data, and this means they have high data demands, and often you need to calibrate them, and those calibrating procedures are quite difficult because you're calibrating over space and over time. And generally speaking, access to this type of program tends to be via the command line. You might need some program experience to be able to use them. So the general picture here is that you require some degree of expertise to be able to run this type of model. And that generally means that often the only people that end up running this type of model are the original model developers. But before I finish, I just wanted to reflect on some work we've been doing in a project called NanoSolve to try and tackle this, basically by putting a graphical user interface, a GUI, on top of the NanoFASE model. And what we're doing here, we're basically taking away a lot of the complexity involved in the model in terms of the data by fixing a geographical scenario and providing lookup tables for our nanomaterial properties. And then so all the user has to do is to come onto the application, which the screenshot is shown here on the right, and they can draw emissions on there. So they can put point emissions on there, which might be things like wastewater treatment plants, or they can draw areas, and that area might represent atmospheric deposition, or it might represent the application of sewage sludge. And then they can run that emission scenario through the model. And then, of course, you can also show the results of the model on the same interface, on the same map. And so really what we've done here is we've taken a complex model that only a handful of people can use and made it easily usable, such that, for instance, regulators could come along and put in their own emission scenario and easily visualize the results from that. And this work is very much work in progress, but hopefully in the not too distant future, there will be a public version of this available for you to go and have a look at. And then finally, just a few conclusions. So today I've described what's quite a complex spatiotemporal nanomaterial exposure model, and given a little bit of information on how you might use that as part of a tiered exposure assessment. These models are traditionally quite difficult to use and required some degree of expertise, but there is at the moment very much a move towards simplifying their interfaces and giving them graphical interfaces to make them accessible to a much broader range of end users. Thank you very much.